My name is John Cushman. I'm a professor here in the University of Nevada, Reno in the Department of Biochemistry and Molecular Biology. We're working on microalgae to serve as feedstocks for biofuel production. One of the major interests in microalgae in particular is that these organisms are anywhere from 10 to 30 times more productive on an annualized basis than terrestrial feedstocks. They also don't compete with food production. They can be grown in non-arable lands. They can also be grown in a wide variety of water conditions. They can be grown in freshwater, but freshwater resources are very limited in the West, so we're also exploring the possibility of growing algae in brackish water and salt water, and also wastewater. One of the major projects that we have in the lab is to characterize algae that are growing in our local wastewater treatment plant. And this is an example of one of the algal isolates. We have a systematic screening program where we then take these isolates and characterize them for desirable feedstock traits such as high lipid content or high starch content. Once we characterize the algae and have it in an actinic culture, we then grow it up in a laboratory scale. And this is an example of one of our laboratory scale flasks. This is a 2-liter flask format. And this allows us to gather data on starch productivity and lipid productivity. After we characterize the algae at that scale, it's important for us to scale up our production. For that, we've built a pilot scale facility in a greenhouse at 2,000 liters. These are raceway pond format growth facilities. We also have an outside demonstration scale pond, raceway pond. It's very important to conduct research on a large-scale format. The reason for this is that just because a laboratory strain performs well at small scale doesn't necessarily mean that that performance will translate into real-world conditions. And one of the advantages of doing a demonstration scale project is that it also allows us to estimate the year-round productivity yields for these different microalgal strains. One of our projects is to adapt species to high concentrations of wastewater, and this is looking very promising, and that is one of the topics that I'm going to be talking about at the upcoming summit. Thank you.